Hi everybody. Sorry I can't be with you today. My son is still not feeling the best, and I got to stay home with him and take care of him. So I thought today I would get us started on the next topic so that when I come back tomorrow, we can maybe play a game and review a little bit about what you learned and hopefully get ourselves ready to do a lab later this week. So we'll get the notes part over with today, and um, and then we can do some more fun stuff later on in the week. Today, we are going to learn about and start talking about how genes are turned into proteins. Proteins are the machines that run your body. Where do those come from? Well, they come from the instructions in your DNA. And your DNA is in every single one of your cells. So today we're going to think about what gets expressed by our genes, where the instructions for making proteins come from, and how do the instructions for making proteins become decoded by the cell. So that's what we're going to get started with today. We'll start with our first note here, which is just kind of a review, where we think about DNA. Okay, DNA contains information. It is only in the cell to do that, contain information, and it stores all the instructions for making proteins. Proteins are the workers. They are the machines. They do all the activities of the cell. So they give the cell shape. They function enzymatically, or they help with enzyme reactions. They help with reproduction. They help with a whole bunch of stuff. Proteins are the workers of your cell. So if the information's in our genes, how do we open it up and read it to make proteins? Well, that process for going from DNA to proteins is actually a rather complicated process, which is why we're spending the entire unit, or at least half of the unit, talking about it. Um, the process of going from DNA to protein contains a lot of different steps, but one of the most important or important molecules that is going to be incorporated in this process of turning DNA instructions into protein machines is RNA. RNA is a little different from DNA. It's similar, but it's a little different. One of the functions of RNA is to carry information from DNA to the cytoplasm so that the ribosomes in the cytoplasm can read it and make proteins. Okay, so we're carrying information from DNA to ribosomes, which is really important. That process of carrying DNA information and turning it into RNA is called transcription. That's what I'm going to try and talk to you about today. As we read a gene on DNA and we turn it into RNA, we're basically writing a complementary strand of whatever the gene is. So if you notice here, our RNA molecule is AUG, UGUGC. That lines up and complements TAC, ACACG. So if we look at this molecule, our gene is right over here. That is, wow, that looks really bad. Let's try that again. That right here is our gene. This would be the complement to the gene, making it mRNA, or messenger RNA. Messenger RNA carries the message of the DNA, and the message of the RNA is only ever one side of the DNA. You also might notice that there is no thymine in RNA. There's no T. The T gets replaced with a U, which is uracil. And uracil is just a different type of nucleotide. It's not really that much different from thymine, but it's only present in RNA, which makes it unique. The last step of protein synthesis, which we will learn about sometime later next week is translation. Translation's a little more complicated, where we turn sections of code, sections of 3 codes, into one amino acid. And each set of 3 codes for one amino acid. Those amino acids, when they link together, create a protein. So that's basically the overview of this process. Transcription plus translation gives you what's called protein synthesis, the making of proteins. And the making of proteins is what we do with our DNA. We turn our DNA molecules into proteins. Or not into proteins. I should say that better. DNA is not turned into protein. DNA is read like an instruction manual. And then workers in the cell assemble the right parts to make a protein based on those instructions. This is what it looks like in the cell. You might be familiar with this based on the board game we played a little while ago. Individual genes in the DNA are read, transcription is done, and RNA is produced. Those were your these were your black cubes. mRNA was your black cubes. That comes out of the nucleus and through translations turned into a protein. What's missing in this diagram is a ribosome. Ribosomes have to take the mRNA and read it and turn that into a protein. If we take a look at the difference between DNA and RNA, we can see some fundamental differences. First, DNA is an information holder. It stays in the nucleus, and it's a pretty, it's a little more resilient than the RNA molecule. RNA molecules get attacked by things in the cell as they leave the nucleus, whereas DNA doesn't have to deal with that as it always stays in the nucleus. There are 3 types of RNA though, so we'll go over each one. There's M, T, and R RNA. mRNA is made in the nucleus during transcription. These were your black cubes. The intermediate, this is the intermediate for making proteins as it carries the DNA message. This is what I was talking to you about earlier. As it leaves the nucleus, it's used in translation by ribosomes. We're going to come back to tRNA and rRNA in a little while. I don't actually feel like we need to go over this yet. I'll throw these slides up here just in case you want to jot these notes down. But tRNA and rRNA are not involved in what we are learning about today. Okay, so we will go over these again, but they're not really useful in what today's discussion is all about. All right, so to finish this up here then, we got transcription. Transcription is the process of making RNA from DNA. All 3 of these different types of RNA molecules can be made from transcription, and the process is the same regardless of what type of RNA you're making. The RNA, the DNA being read and written into RNA language, that is the same process. The enzyme that's going to do this is called RNA polymerase. This is a new one for us. And the location of this process is going to be the nucleus. It's going to happen right there in the nucleus of the cell. There are 3 steps to transcription. We're going to write down each one of these as we go. I'll talk about each one as we get to those parts of the presentation. All right, so let's start with step 1. How does mRNA get made from DNA? First step is initiation. RNA polymerase is going to locate the beginning of the gene and unwind it. The region where the enzyme is attached to is called the promoter site. This is probably the most interesting part of the entire process because it has to do with what you do and what you interact with on a daily basis. So I'm going to go back to this slide here in a second. But I want to focus on this. I said in the previous slide that genes are activated at the, or genes are read at the start, and the start is called the promoter site. Well, the promoter site's interesting because the promoter site always has the same code in the front of it. It's called a TATA box. TATA box is a TATAAA sequence that occurs at the beginning of every gene. The other thing that is at the beginning of every gene are called transcription factors. Transcription factors are elements that your body makes, or things that you eat, or molecules you just come in contact with on a daily basis, or maybe randomly, but some things that your body comes in contact with that can land on your genes and activate those, or at least start a process that allows that to occur. This is really interesting because the portion of where DNA attaches to those proteins that you encounter enhances or suppresses the binding of this RNA polymerase enzyme. So what I'm basically saying here in normal language is that what you eat on a daily basis can activate genes in your cells that would not have been active otherwise. The amount of stress you experience on a daily basis can activate genes that were not already active. The amount of, or the social situations you put yourself in can activate genes that were not already turned on. And that has a big impact on your body and your development as an adult. And that's a really interesting topic, a really interesting process to think about. So when we go back to this first part, we think about, okay, RNA polymerase is going to locate the beginning of a gene and unwind it. How does it know? How does it find the gene that needs to be copied? Well, the molecules you have eaten and encountered on your daily basis, or the molecules your body has generated that are in response to you eating foods or being in social situations. Those molecules land on the promoter site and turn your genes on or off. And if they're turned on, RNA polymerase will land on that gene and start to copy it. That's a really interesting phenomenon, and one that's worth thinking more about. There's a whole field of genetics called epigenetics, and epigenetics is all about the activation and deactivation of genes in your cells. Different cells have different genes that are active, and that's just a really interesting and cool thing to study. So we can review that some more on Tuesday if you have questions about that. Second step is going to be elongation. This one is not as interesting, but it's still necessary. In elongation, RNA polymerase will travel down the DNA. This traveling makes a single strand of RNA that's complementary to the DNA. And while RNA polymerase travels down the DNA template in the 3 to 5 direction, you make DNA in the 5, I don't know why my pen is doing that, the 5 to 3 direction. Maybe that looks cool or something, I don't know. Anyways, don't forget that even though RNA polymerase moves in the 3 to 5 direction, you only ever make DNA or RNA, you only ever make new nucleotides in the 5 to 3 direction. I would really more focus on these bullets or these numbers rather than this bullet up here. All right, termination. The last step in this process is termination, where RNA polymerase hits a termination sequence in the DNA. Typically, this is a bunch of T's. So a series of T's in a row will cause the RNA molecule to be released from the DNA. The DNA will then completely rewind back up. The RNA will move to the cytoplasm, and RNA polymerase moves to the promoter site of another gene and starts to do that same process again. RNA polymerase literally moves from one gene to another, copying all the genes that are turned on. And as they get turned on and off in the cell, RNA polymerase will just copy the ones that are turned on. So now we've got our mRNA molecule. This is our single strand, this is our single piece of mRNA. And that molecule is now going to go into the cytoplasm and be converted into a protein. So that's basically what we saw today, turning one gene into or reading one gene and turning it into an mRNA sequence. I got some practice sequences for you I'd like you to try. Here's your first DNA sequence. Why don't you try writing the RNA sequence right now? I'll give you about 10 seconds. If you'd like, you could just pause the video and do this. Okay, here's the answer. Hopefully you got that right. If you're confused about it, let me know. Here's another one for you. Pause the video and try and do this one on your own. All right, here's the answer. Hopefully you found some success in doing that today. All right, so that's where we were going to end today. What I'd like to remind you of is that you do have a quiz due tonight at midnight on DNA replication. If you have any questions about that process, feel free to reach out to me through email if you have a particular question, or feel free to work together with your classmates on accomplishing the quiz in class today, depending on if you've done it already. So don't forget about the quiz. Second thing I'd like you to try and do, since you didn't really have too much time last week, is to do one more journal entry into your phenology journal. Some of you did not have any growth in your plants yet, and if that's the case, I'd like you to check and make sure you do have some growth in your plants. That would be a good thing to do. And then if you have finished with your plants and your quiz and your presentation or your notes here today, you can feel free to get started on the homework that the sub will pass out to you. That should be done as you work today, either at your desks or your lab tables. So again, I'm sorry I can't be there with you today, but I will hopefully be with you on Tuesday. We can review what you learned today, and yeah, I hope you have a great day. See you soon.